So without wasting time, I would like to welcome all the participants. I'm told it's a very huge number. It's very nice that you have so much of interest or keen interest in learning about the geospatial technologies and its application. Let us say here we are talking about the vegetation carbon cycle or carbon cycle as a whole on the when we talk about the Earth's biosphere. I have been asked to speak on Earth observations and its role in vegetation carbon pool assessment. I shall be sharing my experience of and why we need to do this kind of studies at a national level. There are a lot of papers which have been published not only from India, even from international organizations, but again there are a lot of differences in the assessment. This study which I am going to share with today with all of you, basically the aims that we should have a good basic fresh database of the country, let us say in India. I was coordinating the vegetation part, and the Dr. Patel, he was coordinating the crop part of it. He was part of my team, but he was looking after the carbon pool in the crops, basically agriculture crops, and I was looking in the forest vegetation. That's how we had a very huge team of people. So I'll be sharing it, but I'm told that the participants come from different background. So I will try to initiate the topic by sharing some of the definitions and all that, so that, and then slowly we'll try to justify why it is important. And then how do we do it? How do we do the sampling? And then we will move to how do we use remote sensing and GIS. Even though the title of my talk is Earth observation, but I'm not going to touch Earth observation because that itself is a big topic. I hope, I don't know, all of you, at least a majority of you have some idea about the remote sensing data. If there's any question, I will be very happy to answer in between. Please let me know if you would like to ask any question. So let us start with the carbon pool, which is the today's topic, and we will focus on the vegetation carbon pool. So there are different types of the, let's say 3 types, mainly we can classify the carbon pool. These are basically the reservoirs of carbon, where the carbon is stored, or let us say exchange, anything we can talk about it. So a reservoirs of carbon, basically we can say it is a carbon pool, and we can see this is stored in the vegetation, in the soil, in the litter. When we talk about the terrestrial ecosystems, if you talk about the marine ecosystems, they have much more carbon stored in the marine ecosystem than in the terrestrial ecosystem. But today's focus is basically on the terrestrial ecosystem. And within terrestrial ecosystem, I'm not going to speak on size or litter, but basically on the vegetation. How much of carbon is stored at a given period of time, let us say in 2020, how much of vegetation has carbon stored, let us say last 20, 30 or whatever, thousands of years. We can also say there will be some trees which are very, very old. So basically we are talking about how much of stocking is there, carbon stock. That's basically absolute number we talk about here. Absolute number of carbon, let us say, or quantity held within a specified pool here. So we'll talk about the vegetation. They say roughly 2070 gigatons carbon is available in terrestrial ecosystems. Let me tell you, friends, this number keeps on varying, changing, because as the knowledge improves, the figures also keep on changing. But roughly you can say this much is the carbon pool here. There are 2-3 more terms which are related to the, when we talk about the carbon pool, this term called carbon flux. That's basically exchange. That means a transfer of carbon from one carbon pool to another carbon pool. Let us say when you burn a vegetation or a forest, the whole CO2 or other gases, carbon monoxide or nitrous oxide, all these are released into the atmosphere. So that means it is stored in the trees or plants, and then it gets released. So that's, or it will be also absorbed by the, so that's the sink part of, so release and sink. So when we talk about the flux, that means we are talking about the movement from one to another pulse. And there are units like tons carbon per hectare per year. That's how we quantify it. Now the carbon sink, but today which we are focusing on is the vegetation basically. Because there are two ecosystems which are very important on the earth. One is the terrestrial ecosystem. In terrestrial ecosystem, vegetation plays more important. In vegetation, trees play more important role. So if I say vegetation, let us say any green plant, it can be crop or a naturally occurring plant, herb, shrub or tree, but tree have more important because they can store or sequester a lot of CO2 and store in the form of a timber. Let us say we call it a biomass here when we talk about scientific terms, because timber is a term which is used in the forestry. Foresters, they basically use, or the merchants, they are more interested in the volume of the timber. So they do assessment considering how much of the timber is available. That's called growing stock, and how much can be harvested in next coming 20, 30, 40 years. So that's a different aspect. What we are talking about is today the science part of it. How the sequestration takes place, why we should study, and then how do we use remote sensing in GIS. So that is the basic and today's lecture which I will be focusing on it. I want to be talking about the net primary productivity, let us say. That's the basis, but important topic, but today I think I won't be able to cover that much. Sequestration, that means how the atmospheric carbon is absorbed by the plant through photosynthesis. I think all of us are at some stage or other, either during schooling or maybe in the college days, must have heard about the photosynthesis, where we use the plants use the water, the CO2 and the sunlight to produce food. So that's basically primary productivity, net primary productivity. There are two more terms which I would like to discuss. I mean, either we can say biomass or carbon. Okay. So biomass, let us say, what is a biomass? So bio is, a living. Mass is the weight of it, let us say, expressed as mass. So total amount of living organic matter in trees, let us say, at a given time, if you dry it in an oven, let us say, at certain temperature for certain days, then the water gets evaporated, and then whatever is left is called the biomass. So we can assess the biomass for one tree, one plant, or a group of plants, or entire country. So that's the potential of it. So if we divide this biomass, it has two components basically. The above ground biomass, which we see, that is the stem, branches, leaves, flowers, fruits, that's all above ground biomass. We have equally good amount of biomass stored in the roots as well. When we talk about the terrestrial ecosystem, let us say in the tropics, the roots are not that well developed, whether they are tap root or adventitious roots. But if you go in alpine region, let us say in the Himalaya, beyond 1400 feet or 14,000 feet and all that, you'll find the root system is very well developed. Root biomass is much, much more than the above ground biomass. So when we try to assess, we need to assess above as well as the below ground biomass. Then we have a question, why do we need to dry it out? Please remember, water is a perishable. It gets evaporated, okay, through transpiration or evapotranspiration, the way you want to understand. So this is something which is cannot be stored in nature. It is a cyclic method. So it has to be dried, and after drying, whatever is left called the biomass. Now this assessment of biomass, then we can convert this biomass into carbon. Okay, so we have now good amount of literature available where we can say, okay, if this much is the volume of it, let us say timber or wood, Then there are factors called the specific gravity. It is called specific gravity. That's the gravity of that particular wood. If you multiply the volume with the specific gravity, okay, you get the biomass. I will be detailing it afterwards. And if you have the biomass, if you multiply by 0.475 or 47.5, whatever the parameter you are using, so you can say roughly 50% of the biomass which you are going to get is a carbon. Roughly, okay, and it varies from 30 to 52%. See here, I have written here, there are reports in the world from 32 to 52% of the weight or biomass is carbon. So each species, let us say we have in India, let us say, as far as angiosperms are concerned, let us say we are around 18,000 plus plant species. And if you have to find out the biomass and find out the carbon in each of these species, it's a very big task. It's a very big task, in fact. So people try to do research and try to report these. And then for us, when we are working at a national level or let us say regional level, we can't account for each species actually. So normally we take as a average value, that's 47.5 of the total biomass as a carbon. So now there are three terms I have used here. What we need is a timber or volume, let us say volume of the tree available. If we know the volume, we can multiply with that density of the wood, or we call it specific gravity. Okay, so you can multiply with that, you get a biomass, multiply again with the 47.57 or 4, you can get the 47.57 of the is the carbon of it. So that's how we can, there's another term which you'll find in the literature that's called the phytomass. Okay, this term can also be used very often in the literature. Then the question comes, why do we need to study? I would like to skip some of these slides because it's a little lengthy presentation, but I would like to give you an overview of the entire process of assessing the biomass and carbon. Now, one of the main reasons which we are now facing is the climate change. We are all aware of the potential damage which can do to the existing biodiversity. You see, millions of years also there was a biodiversity of dinosaurs or different type of the conifers, let us say trees and all that. Slowly and slowly, there have been changes and new species have been evolving. So this is just something we can say it's an ongoing process. But now humans are trying to, show their concern, because if human race is lost, let us say at present they are supposed to be the most, race which uses maximum resources of the earth. If climate change happens, then the human race could come under some kind of a threat. So that's the first worry, is that climate change. How the climate change is happening, one of the causes, there are many causes actually, one of the core causes is the land use change. That means cutting of the forest for agriculture purpose or for industries, for infrastructure development, for many, many things, even for urban infrastructure development. So that is the worry, in fact. So our concern is that how to find out how much of the forest we have, so we can use the remote sensing data. I hope many of you might be knowing there is an organization called Forest Survey of India, which does assessment every second year and brings the report how much of forest canopy or cover is available in India. So that's very important contribution of satellite data which FSI is able to make. A lot of carbon is released because of, let us say, nowadays we cannot call them as a sustainable practices of agriculture, or let us say Northeast India we have shifting cultivation, Odisha also we call Podu. So we have this, if you cut the forest and you may grow again 5 to 10 years, but you are releasing so much of it. So maybe in today's context, shifting cultivation may not be, there are different kinds of the views, may not be very good practice actually in view of the climate change. And there are plenty of studies using satellite data. There's a very important study which Flint and Richard had made for the Asian region, and in India also they have part of it. They say a huge amount of forest has been lost, let us in last 100 years or so. Around 50% of woodland has decreased in India. So this itself is a very strong signal or warning that we are, because we are having largest or second largest population in the world, and our agriculture production is there sufficient, but still we need to feed this much of population. So a lot of deforestation keep happening in many areas. Still it is happening if it is not protected. So these are the climate change and land use change because of the population growth. Just look at this diagram. I would like just to remember the gist of it. Let us say we see a lot of CO2 is released. That is the white line in the curve. If you look at it, okay. Now this is from fossil fuels actually. Look at this green line here. This is the emission from the vegetation burning. You can say mainly land use change. You can also say that I might cut a tree, but I can store that wood. I might not burn it. I might store that in the form of, let's say, furniture, or let us say, any form I can store it. But I'm not releasing it. Yes, that's true, but not out of it. So most of it is basically burned and it is released. Either it rots or it burns. Anything can happen. So we see roughly 20 to 24% or 25% of the emissions which are happening, they are originating from the land use change, and in there you have mostly coming from the forest actually. So that is the major concern, and that's where we need to assess periodically how much of the forest cover we have, how much of the vegetation carbon pool is available in country or region or a district or wherever you are doing any study. That's very important input. See, if you do any study at a state level or regional level, it goes, these kind of studies which are based on the original data, they go into the climatic model or global models which people like agencies like IPCC and all that use it. So these kind of assessments are very important for us. You must have seen in the, very often these kind of, when people talk about the global climate change and all that, why we should worry about it actually. Now, if you look at, there are several models, there are several models people are trying to predict. But if you look at the trend of these models, let us say from this is from 2004 to 2010, but if you look at 1860 onwards, also when the industrialization started actually, the trend is like this, you see, it is going up and up. So for some time, let us say it was all right here, but again, it is going up and over. So this is a trend which we need to curb it actually. And that's the major worry for all of us. And if you look at these graphs, how the CO2 emission has been increasing continuously. Even methane has been continuously, concentration has been increasing. So the trend is upward only. If you look at the N2, nitrous oxide is again increasing trend. Sulfur emissions, again increasing trend. Now these are some of the gases which cause global warming. So these are some of the greenhouse gases which are affecting, and the source of these greenhouse gases can come from many things, but we are focusing only on the vegetation part today. Now look at this diagram here. This just tells you what is the balance actually, and why we should try to do the vegetation carbon assessment. Look at this diagram here. Green indicates that this is emission from fossil fuel. So these are the releases, basically the upper half of it. And then you can see the little cyan color, it's net release, okay? Net release here. Okay, because of the land use change. And then you look at the below part, half of it. This is atmospheric accumulation. All that dark brown color you can see, this is atmospheric accumulation. You can see little orange color here. This is oceanic uptake. That means oceans are absorbing. That's what I said in the beginning. Oceans are also very big. It can be source as well as the sink. Okay, it can be bigger than the terrestrial. In the middle, if you look at this part, This we do not know. If this much is released, let us say here releases an X amount, we know about this much, how much of is accumulated in ocean systems and how much is in atmosphere it is increasing. But there's a great amount of it is we do not know anything actually. It is absorbed by, of course, by the plants and trees, but then we do not have the full statistics of it. So that means we need to do some research to know this unidentified sink, which is one of the things you can say unidentified in the vegetation. This diagram is very important for this, called Vostok ice core diagram, which is published in Nature by Vostok. If we just go 400,000 years back, but I want to tell you why we need to study all this thing. Look at the CO2 concentration on this here. Look at this is the temperature rise here. Okay, so two things. We could put in methane also in this thing. They are all interlinked to each other. So if you look at the CO2 concentration and the temperature, you can see what is the trend here. It touches around 280 or 290, okay, ppm, and then comes down. That means glaciation happens, and then after again goes up temperature, again glaciation happens, and this cycle keeps on going. Okay. Now in Holocene, we are somewhere here. You see in the other 280 to 300 ppm, that was the glaciation is to start. Now we are already at around 214 or 220. I don't know today's number. Today it is this much. We are somewhere here actually. That means any moment now glaciation can start, cooling can start. If the cooling starts, the existing biodiversity, existing system, life forms will all go under change. So can we avoid it or can we delay it? I mean, these can be two questions which we can ask. Can we avoid it? Maybe. It's again a big question mark. Or can we delay it? Certainly we can delay it because we know the sources of this thing. Then it is possible to delay it. That much we can do. I have already given you some idea about the factors. Okay. So now we know that in global carbon cycle, and when you talk about the biomass accumulation of the CO2 by the biomass or trees, let us say, and releases, now we know about it. And we know by this time the exact number also. That means it is possible for us to curve this thing. So these factors are known to us now. I mean, you can say land use change or fertilization, nitrogen deposition, climate. There are many, many things which we can talk about it. But then we know most of these things, so we can do it. So what is nowadays? Now people are focusing or showing their concern on the production forestry, how to reduce atmospheric carbon through forestry. That means the trees can absorb or sequester atmospheric carbon through photosynthesis and can hold it for many, many years. In fact, thousands of years they can hold it. There's no in that. You must have seen if you go to the some part of the Northeast India, maybe in the Western Ghats also, you can see huge, huge trees, giant trees, where you need to 4 or 5 people, you need to, you want to measure the diameter of it, you can't embrace, you need 2-3 persons to embrace. So you have the mother trees or very old trees growing still in our country. Let me tell you, in Arunachal Pradesh, if you go, you can still find huge, huge trees. Not of course like the sequoia and all that which you get in other countries, but we also have conifers which are gigantic actually. And therefore it is very important that how to decrease the atmospherics, and then how much has decreased also is important. Who is decreasing is also important. So vegetation is important to assess it. There are many reports, let us say second assessment, third assessment, fourth assessment. People are talking about the global IPCC is involved in this kind of, they're releasing every year what is going to happen if climate change happens. So assessment of the vegetation carbon becomes very important for us, let us say, when we work at a local level as well. So now, see, every government, now we are committed to do some kind of a assessment actually, because we have to report to the international bodies. So we would like to study how much of carbon is available. That's one thing. So that can be done. People have done it. I will show you. People have done it, and there is a huge difference. And the assessment done at different times. Same person, same team, let us say, not person, let us say same team doing at time 1, the number comes different. Time 2, number comes difference. That means the approaches are now evolving. We know where are the errors. That's why we are trying to improve the number which we, let us say, have in our country, let's say in India. That's the one of them. There's another thing, let us say, if you look at those who are from forestry background, they would know that India has around 221 types of forest. It's a huge, huge number. You don't find anywhere in the world. 221 types and subtypes of the vegetation or forests are found in India. It's a huge number. You talk about the southern part of the country, from mangrove vegetation to the Himalayan region, alpine vegetation. And then you have in the west, hot desert, cold desert, and you go to the Meghalaya, or you go to Kerala, let us say, where you have highest rainfall areas. Okay. So that means we have so much of variability. Then we should know what is the potential of carbon sequestration of these 221 types of the forest. Do we have that number? We do not have, in fact. We have for major class, let me be sal forest, teak forest, or let's say dry deciduous forest, or moist deciduous forest, or evergreen forest. We may have some numbers, but we do not have type-wise number. What is the rate of carbon sequestration or photosynthetic efficiency, or what is the rate of primary productivity, let us say, or net primary productivity, or ecosystem productivity. There are many things in that. That means there's a need to study this. And why we are doing, emphasize again, it's just we would like to mitigate the climate change, because we have realized the mistake which we are doing now. Many countries are concerned about it. Some are really doing something good, some are not very much. So it is important for us to study how much of national contributions are there, how much source and sinks of CO2. It is important for us to study. Then we need to evaluate the different processes which control CO2 accumulation. That's the primary productivity literacy. We need to try to do the terrestrial as well as aquatic carbon sinks, how much are there and how much is available there. And then we can do what we call the carbon budgeting for the region, we can do for the country, and that stats can be used for the budgeting or carbon budgeting in the entire globe. So there is a possibility. Let me also share with you all, friends, carbon is not bad. Please remember, if there is no carbon, there will be no heating. So we need to have certain temperature to be maintained. Otherwise, there will be all snow everywhere. Then life will be different. Of course, there will be life, but it will be of different form. So carbon is not bad. Our body also has carbon. But excess of carbon in the atmosphere has become a major worry. Because when the carbon monoxide or carbon dioxide or these gases increase in the atmosphere, then the global warming, they don't allow this solar radiation of high, longer wavelength, let us say, to pass through. They block it. And that's how the global warming is happening. And then we can think about it, how to, overcome this problem. Let me also tell you that in the past, let us say millions of years, the study says that CO2 levels were much more than at present. This is not the first time in human race, let us say, age here, we have this issue. In the past also, if you look at the evolutionary history of the Earth, many extinctions have happened, many, not 1, 2, or 3. Okay, sometimes CO2 goes up, sometimes oxygen goes up, sometimes carbon goes up, sometimes methane goes up, and then it balances, again it starts, that kind of cyclic or non-cyclic events are going on. So let us see where we are in India, where we are. So total land area, let us say we have around 2.4% of the share. So we are 7th in rank area-wise. Population-wise, we are second number. Cattle livestock, I think maybe first or second. I was listening to some news, they said we are second. Maybe my slide may be old, but I think maybe first or second. Arable land, we are second. Irrigated land, can you, such a fantastic irrigation system we have, that we are the first actually. And when it comes to the forest and woodland available, don't worry about this number 67.55 million hectares. Okay, this keeps on changing. There will be some, definition of the forest has been changed. So there is some increase here. Okay, so we are 15th, let us say, in the as far as the forest cover is concerned. So that's the overall view. So when it comes to the human consumption, or let us say livestock, it's we are, top of the world, you can say in one way. Land area is very less, forest area is very less. So we have to do something for it. And that's where the carbon sequestration we need to study. And one of the primary requirements, because now we are talking about the IPC rules and regulations, you must have about the Paris Agreement also, sustainable development. You must have also heard about the different kinds of the resolutions which have been taken. There are many things which are you can find in the literature. But then what is important for us is to do a study at a local level, so that your data, your own observations, when you publish it, they become input for global level or regional level monitoring or modeling. So we must try to study how much of the CO2 is absorbed, okay, how much is captured, how much is released. We need to know about the balance of it. So nowadays, like the different mechanism, like clean development mechanism, agroforestry, let us say in India, we have agroforestry, social forestry. There are different schemes are already going on, eco-development. Clean development mechanism is one of that people are encouraged to grow trees, and then they are compensated after few. There are conditions and all that. I'm not going to detail of this idiom. Just an overview of what are those ecosystems which are very productive, let us say, which have a lot of potential to sequester or are holding a lot of carbon. Okay, I don't want you to remember the facts and figures which are mentioned in this slide. Just what I'm trying to say, at high, let us say from 50 to 75 north latitude, let us say high, and mid latitude, the Singh, see, you can see a lot of carbon sink is around 0.47. Okay, so per year, roughly 0.7 plus minus error here, gigatons per year carbon they are sequestering in mid latitude and higher. So not very high, 50 to 70. Low latitude, let us say 25, 0 to 25, let us say from equator to 25, around 52%. Okay. Similarly, release also is available from fossils, let us say in 1990. Now it figure must have changed now, 6 gigatons carbon in 1990. What I'm trying to tell you that there is a possibility to improve these numbers if we have studies at local level. And the study which I'm going to share with you, this methodology and all that, it's only for India we can do it. In the beginning, I told you that there are several studies. It is not that we are trying to emphasize or do this kind of study for the first time. There are many studies which have been carried out. Let us say if you look at this from 1980 to 1995 and our project which we carried out from 2004 to 2012 and all that. You see, they have forest area they have recorded here, and they have assessed the, let us say, phytomass carbon pool, let's say in teragrams per carbon. Okay, 2,587, let us say this is Hingane. Then Dadwal et al, this team, then they assessed 3,107. And then Dadwal and Shah again increased number. Within 2 years, they revised the number. In 94, again you see, okay, again here they bring down this number in 85. And another study, let's say Ravindranath from ISCC and all that, Bangalore. In 86, they assessed this much of the carbon pool is assessment in country. Chhabra et al, again, this is a team of Dr. Dadwal. They assess this much. Lal Singh and all, they again, this. So you can see within a span of, let us say, 15 to 20 years, several studies have been carried out. But this carbon pool assessment, which we say is not same. There's a huge difference, almost 40% to 50% difference in the assessment. Why this difference is there? There are several reasons for it. First reason is that most of these assessments are based on the observations made by the forest departments of, let us say, in India. So forest department, they do the forest inventory. They collect the information of the diameter, the tree height, the species. If you know the diameter of a plant, we can find out the volume of that tree. If you know the volume of tree, we can find out the biomass. If you know the biomass, we can find out the carbon. So that's a very simple method which they have been. But same data, different approaches. So somebody uses, let us say, ecological studies from the two forest type density wise, they can do it. FOO has its own inventory system. State-wise, they have done the growing stock. They took and then they use the biomass expansion factor, BEF. Okay, this is very important factor because let me tell you what happens. No, the timber, or say merchantable timber or commercial timber, talks only about the bole. It doesn't talk about the finer branches, secondary, tertiary, and onwards branches. No, primary branch, if it's a thick, they may consider as a timber. So that means many things are not accounted. So we need to have a factor to know the entire biomass, not only above ground biomass or above ground carbon, even below ground carbon also we need to study that also. So factors have been developed. So that means there's an improvement now once you use the factor. You again see the growing stock based. Then comes the remote sensing use in between. Earlier also remote sensing was used. And we are using most of the time the photographs, aerial photographs for forest type mapping and all that. Now again it depends. What is the level of the mapping which we are doing? What is the scale of mapping which we are doing? What is the sampling strategy which we are following? Each one of is important because it all depends how the data has been collected and processed. So that's why these differences are there. So that's the idea to tell you why we need to assess again and again. There's another thing, let me tell you. Each country is supposed to inform to the let us say IPCC and the other international organizations. It's called NETCOM, National Communication. NETCOM means National Communication. That basically is how much of carbon is stored, let us say, in vegetation in India or in any country. That's called national communication. So Ministry of Environment and Forest and Climate Change is supposed to assess and inform that this much of is available. But then there are two aspects here. One, inventory which is done by administrators, let us say forest department. They have their own traditional way of doing. The other approach is for researchers, let us say people like we who do more of, let us say, go into more details of the research. And then these methodologies are shared with those managers or administrators so they can improve their assessment. And that's why what we are talking now about is that we need to understand this cycle much better than what we have. And therefore, it is important for us to measure or determine the biomass, carbon pool, growth, and yield. So let us get 4 things. We should know the biomass available, how much of the carbon pool or reservoirs is available in our country, what is the growth, let us say, what is the increment in the diameter, increment in the timber, increment in the biomass, increment in the carbon sequestration. So let us say with X rate it is increasing, but then with X rate it is being cut also. How? The forests are, trees are being cut for timber. Then a lot of fuel wood collection is happening day and night. A lot of grazing happens so a lot of things are already are taken out of that pool. So we need to know the balance of it. And then we should also have the idea what is the yield after 10, 20, 30 years, how much of yield is going or how much is we are going to have the store, let us say. We need to know that. And therefore people are now trying to justify this for forest management. If we know that, okay, we have less, then we must do the more afforestation, reforestation program. CDM is one of them. The Green India Mission is one of them. There are so many schemes like Bihar. We have now, what is that? Some name is there for forestation. Okay, so revenue is one of the things. Conservation is one of the most important thing. We have to do that. And then what will be the demand and supply in the future? We should know. And that's where the assessments are very important. So how do we go about it actually? So let me go into a little more about the methodology part of it. It's very important or very easy, let me tell you, to assess the basal area. Let us say we have a tree. What is the basal? If I know the diameter of it actually, it's possible for me to find out what is the basal area. Now basal area, height, diameter, volume, carbon, biomass, these are all correlated to each other. If I know the even canopy diameter, the tree canopy, if I know the canopy perimeter or let us say dimension of, I can correlate or I can find out with the tree diameters, let us say DBH. So the DBH is very important, diameter at breast height. If I know the diameter at breast height, I can do many different kind of assessments. So it's very important for us to know the volume of the logs which are harvested or which can be harvested. It's very important for us to know the total growing stock or harvestable stock, because growing stock will have mature trees also. Now, the mature trees, it's good to harvest them. We should not be against harvesting, but we should know how to harvest, what is to be harvested. If a tree has become old, let us say very old, it is not growing anymore. If it is not doing any photosynthesis, it is just surviving. It is better to harvest so that that space which is occupying receives more sunlight. Then it will allow other plants, the sapling and seedlings which are below the trees or below the forest, to start grow faster actually. That means we are increasing the regeneration of the forest. So, I mean, it has to be managed. We can't recommend that clear felling. Okay, you cut everything and restart. That's not the good practice. But if we know that tree has matured, this is the age of, there are different methods to do that in forestry. We do have methods of cuts and all that. We can harvest it. So that's also we should know about. And as I said earlier, height, volume, age, all these are correlated to each other. We can use now remote sensing. We can bring this data into GIS domain for analysis. So let me give you the overall methodology, how we are going to give in details of it. To start with the, let's say, carbon assessment or carbon pool assessment, I said biomass assessment or timber or volume assessment is the first requirement. But before that, you should use remote sensing data and should have a vegetation cover type map. That's the first thing. We should know where is the carbon available or where is the carbon stored by the forest or trees. Where is that? First thing we should know. Second is, what is the density of that? Because if density is high, that means high stocking, high amount of carbon, high amount of biomass is present. If it is open forest, more of grasses, less of trees, that means less biomass or less carbon is present. So satellite data is used nowadays. You can use Landsat data. It all depends which level you are working. Suppose you are working at a country level, maybe you have to go to AWIS data or MODIS data. But if you're working at a state level, I think Landsat data, IIRS, these three data, you can use it for mapping. If you're working at district level, let us say, or still lower level, let us say block level, we have high to very high resolution data of ICONOS. We have our own data, LIS4 sensor we have is very good data. Cartosat, if you want to further down, you can do Cartosat data. So very high resolution data is also available for all this kind of mapping, which I'm not discussing here, but I'm just telling you what is the role of remote sensing. So the role of remote sensing starts for any assessment. Is role of remote sensing comes in the beginning and at the end. Beginning is the vegetation cover type mapping, density mapping. Ending is the modeling, spectral modeling, how to do the geospatial modeling of these observations which you have taken in GIS domain. So that means we have remote sensing, GIS field, everything has to be integrated. That's what I'm going to discuss with you. So first thing we should do, satellite data helps us to do the stratification in the form of cover type and density. So forest, I'm using the word cover type, not the forest type. Please remember, Forest type means if a vegetation type is based on the field observation. When you go to the field, you know what is the dominant tree species, co-dominant tree species, and what is the understory vegetation. That is based on the field observations. When you describe a type, that's called the vegetation type or forest type. But in remote sensing, we don't do that. We see the remote sensing from the, let us say, from the space. And what we see is the canopy cover, tree canopy only. We don't see the stem or the pole of it. So that's why it is called cover type. And each forest type will have a different tone, texture. I think maybe I will tell you what is that. So there are different types, different colors, different density, different tonal variations. Okay, so texture variations. So density is related to texture. Tone is mostly related to the overall. I'm telling you, there will be some exceptions overall in the forest type. So starting the remote sensing, we do the stratification. Once we have done the stratification, we can now go for sampling design. Where should I do the sampling? What should be my sample size? What should be my plot size? How many plots I should take? All that now we can calculate based on these two maps which we have prepared from the satellite data. Now let us say we have now collected the field data based on certain sampling design. Then we need to calculate it. That's third, fourth step you can say. For that we need to have a called site specific, species specific volume equations. Please remember, site specific, species specific, volume or biomass equations, because what we are trying to do here, biomass and carbon assessment. So either I should have a volume equation, volumetric equation, or any other allometric equation which is available. Nowadays, people are not developing biomass equation directly actually. Okay, you can also develop. I would urge the participant, those who are interested actually, please develop biomass equations or volume equations for your area where you are working. Ask your students, take their help. Because what we have equations are national level. They are based on certain kind of inventory. Okay, but at the time when these equations were created or generated, the objective was different. Now objective is different actually. So please do that. And then we will do the regression modeling, geospatial modeling of biomass and the carbon. We sometimes even one season data is good enough. One season NDVI data or AVI data or AVI or spectral modeling is good enough. Many times it's not good. So it is better to do the modeling using multi-season data. That means I have a, let's say, summer season, growing season, or let's say winter, or let us say just before rainy season. There, our country has, let us say, 6 type of seasons. We can have the multi-season data available. And that means once I have this, that I can generate the coefficients. That means I can relate the pixels. Now we have the spectral modeling. So I know that plot size, what is the biomass and carbon present in that plot, in that pixel. I can correlate, develop a model, simple linear model or polynomial model you can develop, and then you will be able to get a coefficients. Equation is now available. Just put the independent variable, you get the biomass or volume or carbon, whatever the equation you have. So that's how you can take it to the regional level actually. Just to give you again some idea about above ground biomass, we have trees. In trees, let us say what are the different parts of the tree: bole, branches, twigs, leaves, flowers, fruits. They're all part of the. So that's one plant form or habit. Shrub is another habit of it. It has the stems. Let's say tillers. We call here tillers because they don't form really big stem. There are so many branches. We call them as tillers. Branches, leaves, flowers, fruits. In herbaceous plants, those grasses or climbers or epiphytes or lithophytes or whatever the plants or forms are there, we can do the whole plant collection or part of it we can collect and then we can assess it. There's a lot of debris which is lying on the floors of the forest. That also is contain biomass and the carbon. So your debris can be in two forms. It can be litter or humus. Litter is maybe fresh or little old, not rotten basically. Humus means it is rotten. The leaves have become brittle, black, soft. If you break it, they will become powder. That's called the humus. So above ground, the biomass or carbon is available in these 4 or 5 forms, which we can assess. Below ground, we have the roots of maybe trees, shrubs, or herbaceous. Okay, and we are not talking about the nematodes, animals, protozoans, bacteria here. Okay, but they are below ground that is also available, but we are not focusing on that. But my today's lecture is basically focused on the above ground biomass. So what type of equations are available with us? Okay, those who are from forestry background, they would know about it. It's a very simple linear equation. Okay, y is equal to a plus b or alpha or beta, anything you can use it, into x. So y is, let us say, your volume or biomass or carbon, depends upon what equation you are using. Alpha and beta are your constants. X is your independent variable. Here independent variable is what? Diameter of the tree or height of the tree or both of them, diameter and height. That's all. We don't need. Now, of course, there are some new studies. They have tried to correlate with the canopy also. Because from satellite data, we are getting only canopy. We are not getting the stem diameter. So if I can correlate the biomass with the canopy of it, I can use the canopy reflectance as an independent variable and try to assess this. So these are, a lot of research is possible, a lot of variations are there, a lot of research is possible here. So once I have the first thing, let us say I have the volume. Volume can be multiplied by the specific gravity or wood density, we call it. It is available in the literature. May not be available also. Here again, let me tell you, every time I'm emphasizing species specific. Let me give you a simple example. Teak growing, let us say in the southern part of the country, or let us say Nalla Manlai or other parts of in the southern part of the country, the rate of growth, the climatic conditions where they are growing are different from, let us say, central India and Madhya Pradesh, okay, or let us say in the other parts in the Uttar Pradesh, some part of it. That means from very moist conditions to very semi-arid to arid conditions. So rate of growth will be different. The specific gradient, wood density also will be different. Let me also tell you, wood density also varies within a tree. In the same tree, if you take a main stem, it is different. If you take a branches, it will be different. Ultimate branch, it will be again different. So what we take a mean of it actually. So now let us say I have the volume multiplied by the specific gravity, I can find out the biomass. And if I know the biomass, I can multiply by just 4.47. That's the percent of the carbon which is present in most. This is a generalized number. This is an average number, please. I told you it varies from 30 to 52%. So if you're working at a national level, maybe this is good enough. But if you're working at district level, maybe it will be good for you to know the percent of each of these species which you are working on it. Then you can have a better assessment. Okay, so we can now find out the carbon out of it. Okay, so there are several studies which are available. What I'm trying to say here, there are early days, let us say 1970s and all that, or let us say before that also. I mean, aerial photographs were used for mapping and inventory, and trees were observations were used for assessment. And slowly what happened, no, we came into the remote sensing part of it. You see here, remote sensing started. So now we go to pixel level. Some of the pioneer works which are Tiwari and Singh, from the ISRO, Kankawell, they are some of the pioneers who have used the aerial photographs or IIRS data or any other data to bring or to correlate these reflectance at pixel level with the field observation and try to generate some equations. And of course, now we have the models, GIS based and all that. So a lot of progress has been made in this thing. Let me give you now the project which we talked in the beginning. We had a national carbon project actually, which was headed by Dr. V.K. Dadwal. I had the opportunity to coordinate the vegetation carbon pool of it. That's the Abhishek told in the introductory message. What we did actually, see, one of the things which we found, at that moment was that the sampling strategy followed by Forest Survey of Forest Departments, let us say Forest Survey of India, State Forest Department, FSI, FRI, ICFRI, okay, they go for 0.1 hectare plot. They have a standard. There's no doubt about it. Researchers, no, university researchers, let us say academicians, they have their own methodology. Somebody will say I will take 20 by 20 meters. Somebody will say I will take 100 by 100 meter. Within that, I will do the subplotting. So in this project, we try to put things in an order so that we have a uniform methodology followed in entire country. So what we did actually, so we had a forest type map from Forest Survey of India. We had from the biodiversity project, we had the forest type map. Forest density map is readily available from the Forest Survey of India. NDVI you can get within 1 minute or 2 minutes. Road map is also available for accessibility. So we, using these 4 or 5 parameters, we identified around 22,000 points, sample locations in the country. And out of that, there were 4 actually. This is a different methodology which we are using. And we gave to, as it said, there were 70 PIs working from different universities. I must thank every of the collaborators from different systems available. They contributed in this data collection. We could do around 7,900 or 8,000 something plots we have laid. We had extra plots because sometimes you are not able to reach that plot. So same density class, same NDVI, same forest type available elsewhere, which is easily accessible, can be taken. So, I mean, we have to optimize on money, manpower, and the time. And that's remote sensing gives you great advantage. You have an NDVI, so you know the NDVI range. NDVI range is used for quantification of biomass. So if you can have 10 classes as a point, so let us say less than 0.1 to 0.1 to 2 and all that, you can divide it. And in each NDVI class, you can ensure that I will take measurements. Then when you try to develop a model, NDVI versus or spectral modeling with any ground-based observation, you are likely to get best observations, best results. So we identified these plots in the country and gave to the collaborators to do the data collection. I have already told you about the volume. How do we get? So biomass expansion factor. Further research can be done. I'm just telling you not only the what is being done, what you can do is component-wise biomass assessment. You know, I mean, Dr. Tiwari in 1984 and 85, they proposed this idea of it actually, but somehow he, you know, untimely demise of him, this work could not carry out. But we have now worked out more on that in this project, that how to collect component-wise data and assess the biomass and carbon of each of the, let's say, main stem, bole we can call it, then primary branches, secondary branches, tertiary branches, and so on, and go up to the last branch which bears leaves and the fruits. Now we will have, see, the earlier volume equations which are only using the main timber, commercial part of it. But when you do the science, we are not only worried about that, we are also worried about the other parts of it. So I would suggest those who are interested should carry forward this component-wise assessment. Even if you can do it for one tree in 1 year, is a great achievement. If you can do it sincerely and publish it, okay? So now these things are very important. What are different methods of data collection? There are basically 2. I can destructive method. That means I can cut a tree, dry it, take a weight of it. Okay, that's one part. But nowadays you can't cut a tree because already we are short of forest cover. So further deforestation is not allowed. But for research purpose, yes. Now what you can do is that you can, in area where you are working, you can do the girth class distribution, like let's say less than 30, 30 to 60. And in each girth class, if you get a permission from the forest department, you can harvest one tree What for? To develop a equation. This also will not be allowed because it's not, because now better methods are available. What I would suggest you, during cyclones and all that, no, some wind storms, many trees get uprooted. It's a golden opportunity for you to do all the kinds of measurement. If you can get in touch with those authorities who are the owners of that, take measurements, take pieces of it, and do the research. So nowadays, road widening is also going on in the country. A lot of trees are cut around. So locally, you can do all this kind of research. Okay, that's what I would wish. Full tree, you can't weight it. So we have to do the some kind of measurements and then try to develop equations and try to. So descriptive method is not advisable, but maybe sometimes if you get a permission, yes, you can do it. But I would suggest not to do any, because we have non-destructive methods also available. How do we do the destructive methods? Let me tell you. Okay, let's say on right side, this is a forest of, this is a Deodar forest, okay, in near Manali, if you go. And let us say this is a tree, of course not the same tree, this is a tree in the Coorg district in the Karnataka, okay. What I'm trying to show here, we can see, we take the diameter at breast height, okay. Now what I do, I take a slice of stem from this part, lower, at certain height, I take this slice, I take this slice, I take this slice. So I will be able to find out the relationship. So I know the diameter of it, I know the weight of it, I can calculate for every piece of wood which I'm collecting, I can collect the, get the biomass, the carbon, and then I can develop the equation. Tree height is known to me, you can measure the tree height, diameter is known to you, the canopy cover is known to you, everything is known to you, you can do it actually. So this is one methodology which people do it, and you can see here, you can see a person here climbing. You can see there's a clean bowl, but this person could climb here. Can you imagine the kind of strength he has? So it is smooth, he has climbed. What we want out of him is to collect the information on these branches. What's the diameter of branch? What the length of this branch? Because we would like to also assess the, connect it again. So we were talking about the two methods, destructive and non-destructive methods. As I said, destructive methods are not supposed to be followed these days. Government also will not allow you, but if some trees are falling for some reason or being cut for some reason or other, you can get opportunity for you to do that. What I was telling, no, that if suppose a tree has fallen in your area or is being cut for road widening, what you can do, you can take slices at different height. Let us, this is 1.37 meter height. This may be at 5 meter height. This may be at 10 meter height. This may be at 20 meter height. This may be at 25 meter height. You are taking the disc. You can take a saw, take a slice. So you know the diameter of it. You know the width of it. You can weight it. So you know the fresh weight of it. You dry this in the oven. So you can find out what is the biomass of it. So if I know that this diameter, this height has this much biomass, and then I can correlate all these things. Okay, so that's how this field work is carried out. You can say semi-destructive or non-destructive way. This is one example which I was giving you, that you can't cut a tree, but yes, some tree has fallen for some reason or other. You can do this kind of sampling. You don't have to carry whole tree and dry it and weight it. No, just take slices in between and then try to generate a model out of it. In the semi-destructive, what I said that here we either no destruction or semi-destructive. Semi means, okay, little bit. See, now we, as I said earlier, we call it a borer, tree borer. Okay, the borer is inserted into the tree and you rotate it. It needs little of strength, a lot of strength actually, to pierce the borer into the stem up to the pith center part of it. Take it out, you will get a wood piece in it, maybe 10 centimeter, 20 centimeter, 30 centimeter. You know the length of it, you know the diameter of it, it's known because we know the whole diameter. Then I can take fresh weight of it, I can dry it in oven, I can find out the dry weight of it. So now what I do, I know the volume of that piece of wood. I know the biomass of that wood. I can also find out the specific gravity using the same sample. If I know all these parameters, that this much of volume has this much of biomass or this much of carbon, then entire tree, let us say, what will be the biomass? I can extrapolate this thing information. But then what I need to have these observations, either little bit of sample of wood, even if nothing is available. See, that's what I'm trying to say here, that whenever you do this kind of study, the growing conditions are different. Teak or sal which grows in Dehradun, the rate of growth is different. The sal which grows in Rajgir Hills, where I am staying nowadays, in the Nalanda University, is very thin. Very thin. Site conditions are not poor, maybe 30 years old, but diameter is very less. Whereas the same sal, suppose it grows in the, let's say in the Shivalik Hills, grows much faster actually. So the volume equations, biomass equation is specific, they have to be site specific. That means for UP it will be different, for Himachal it will be different, for Uttarakhand it will be different. So we can use the satellite data for type and density mapping. We can overlay these to prepare a homogeneity map. Homogeneity map basically means uniform. So forest type, one density, type, density, and the physiography or any other parameter, or you can take aspect also. So northern aspect, the same forests, okay, this sample plot I will take. And then I design, sampling design, I'm going to tell you a little bit about that. Take measurements on DBH height, crown and all that. And then you have the species equations available, so you can find out the volume. And then bring into remote sensing data for the, see, that's what I'm saying, GPS is important. And before you go to the field, please try to identify in which pixel you are going to lay your plot. That means your geometric correction of your satellite data has to be as accurate as possible, and put your cursor in the middle of it, note down the GPS latitude, longitude, use a GPS to reach to that location and then lay a plot. So what will happen? You know the plot dimensions, you know the pixel dimensions, you know the spectral reflectance in green, red or infrared or middle infrared band, you know the biomass per pixel, now you can calculate, you can correlate. So you can develop a model for extrapolation further, because you are doing sampling for a small area, but you have to do mapping for the entire district or state or country. For that you need geospatial modeling. Okay, these were some conditions which I was telling, because they're important. We do not have every time enough funds available. So first thing, please decide what you want to do. What is your objective of, let us say you want to study biomass and carbon. Okay, fine. How much area you have to, how much time you have, how much money you have, and how many people you have in your team. And let me also tell you, what is important for you is when you go to the field, someone should know the name of the species. That's very important. Many times we take the villagers along with us to the field, they tell us the local name, that's good enough. So we can find out the from local name or vernacular name, the botanical name, because botanical name is important to find out the regression equation or the volume equation. Otherwise, it's very difficult. So you need a good time spent identification of the species, do the stratification, do good sampling design. There's an equation for that, I'm telling you the equation also. And then use satellite data. Nowadays, aerial photographs are not available. You can use very high resolution satellite data. Even Google Earth is good enough, let me tell you. Even Google Earth is good enough for you to do your sampling design. Just take out that area and do it. And you can also use see, FSI is publishing type map and density map. ISRO has also vegetation cover type map. You can use straight way, it is available. You can use straight way for type map. Density map you can take from FSI. Just put over the other, you can prepare a homogeneity map. Okay, so what kind of observations have to be taken? So diameter is one of them. Diameter at breast height, that's normally in India we take at 1.37 meter, okay, here. 1 meter, 30 centimeter, 37 centimeter, that's called breast height basically, okay. At that height the diameter is taken. In India it is followed. In some countries it is 1.6 3 only. That means 130 centimeters. We use 137 centimeters. You can use it. Again, in forestry, they take observations more than 10 centimeter diameter of the trees. But for us researchers, I think more than 3 centimeter we should take observation, because up 10 centimeters is a good diameter actually. They also have biomass, they also have carbon actually. So why should we miss it? We should observe it actually. We should have observations on the tree height, clear board height, how much is there, mean height of the plot, because if you want to relate with the ground cover, canopy density, crown area, what is the shrub cover, because even though the biomass of shrub and herb is very less, maybe less than 0.001%, but when you do a scientific study and if you say I have done only for trees, okay, people will say, no, you have not done for shrubs, you have not done for herbs. But one should know that the amount of carbon or biomass of shrubs or contribution is very, very less. You can ignore it also, but it becomes incomplete studies. So these are the few parameters which you have to collect from the field. And I would suggest those who are interested should have a good format designed for how to collect that. Then choose a good satellite data for your study, high, medium, or coarse resolution data. So I would suggest you go for medium resolution data if you're working for district level or state level. Coarse resolution data, let's say MODIS or and all that, no. You will get something, but I would suggest those days have gone because now we have a better resolution data. Why not to use that one? Always go for a multi-season data. See, let me tell you why we should go for multi-season data. Just take a simple example of 2 important trees which grow in India, teak and sal. If you try to map during the rainy season, okay, every tree has leaves, so you will not be able to say which is deciduous and which is evergreen. So season selection is very important for mapping and for modeling also. It's very important. So I would suggest please take multi-season data, and wherever you find very high R-square value, okay, take that data for further modeling. That's all, very simple. It's not, it takes time, but that's the way to decide on that. And then of course we use the volume and volumic biomass equations, specific gravity or wood density to find out that. So I was telling about this methodology, probably there was some problem. Let me summarize what I have been telling you so far. So we start with the satellite data. We do the forest type mapping, forest density mapping. We can take NDVI from the satellite data. We can take road map, accessibility, and we can also overlay district boundary on it. So now this will help me to decide the sampling design. Then I will find out for each of the location, latitude, longitude, give that latitude, longitude to my team. They go there and lay that plot in a homogeneous strata. Okay, this is homogeneous strata is coming from here. Now within this, we will do the cluster sampling. That means at one location of, let us say, MODIS data 250 meter by 250 meter. Okay, in this study which we have followed, we can lay 4 plots of 0.1 hectare. And then why 4? Because then, see, when you go in a forest type may be same. Even density also, if you have a very broad class, it may be same. But local conditions will be different. Local climatic conditions will be different. So it is better to take more plots from the same area so that all small micro variability, small variation in terms of species, site conditions can be accounted for it. That's why this is better. Let us say I have now the field information. I now create a database in MS Excel or any spreadsheet. Okay, I calculate the let's say biomass or volume, whatever I want, whatever equation I have. If I can calculate basal area and the volume multiplied with this specific gravity here, I can calculate the biomass, tree level biomass, and I can sum up, I can find out the plot level biomass. Then I can convert this plot level biomass to per pixel biomass. Very important, please remember. Why per pixel? Because we are going to do the modeling using satellite data. So pixel versus biomass, they should match. And we can do the geospatial modeling for the entire year. In between, there are 2-3 exercises which you can think. You can try to check the error actually. Because when we do the field observations, the field conditions are not very, very helpful. There could be some error in typing or writing or noting down. So how to check those errors? So there's a possibility to check all that. I'll show you some graphs of that. This is a little complicated methodology for the plants which are less than 10 centimeter in diameter. This is a methodology which is more than 10 centimeter. This is less than 10. It is very complicated, so I will skip at this moment. But if somebody is interested, can go through publications of mine. The whole methodology is explained there actually. Now I have, let us say, a map with me. I should have proper season data, proper spatial resolution data. It all depends upon your objective, and proper spectral resolution from multispectral to hyperspectral. You have to decide it. I'm focusing only on in optical region. I'm not talking about the microwave region or LiDAR at this moment. Okay. And as usual, we do the radiometric distortion removal, geometric distortion removal, and we do the enhancement. These are the basic things in remote sensing. I hope those who are, and those do the visual interpretation or digital interpretation. Okay, that's how the map starts. Let us say I have this kind of a map. Okay, a classified map. This is a part of Doon Valley, Sal Forest. So I can say all this Sal Forest, Sal plus Roni, Lantana camera, Syzygium Camini, dry deciduous teak, Sal, all these. Let us say I have a map of this kind of your area. And then I have different density classes. See, this is the type map. Then I can have a density class also. So this is, let's say, more than 80%, 60 to 80%, 40 to 60%, 20 to 40%, and less than 20%. You, in your area, you will have to decide what was density classes present in your area. Okay, it will vary place to place, but we should have this kind of a map. Then we can prepare a homogeneity map. How it is prepared? So this is, let us say, a type map, Sal type map. This is a density map. Right side is a density map. This is a Sal map. Now Sal, this, if I overlay these two, okay, in GIS, I get this bottom one. So I get a forest type wise density map if I overlay. Now these forest type wise density, they are the homogeneity. This is a homogeneity map. These are homogeneous areas. In each homogeneous area, if you can note here, there are round bullets are there. These are the locations which we have identified to do this sampling. So not even a single strata will be left out. So we will be touch upon all the stratum which are or strata which are available in that particular area. So this is very significant to prepare a homogeneity map based on the, you can use type and density. If you have physiography map, there's also you can. So that's how we have done it here. We have found out the area of each class. That is pure Sal 10 to 40%, pure Sal 40 to 60% density, pure Sal 60 to 80% density, pure Sal more than 80%. Forest type remains the same, but density changes, area changes. Similarly, number of plots also changes. That's very important because if you have less sampling or over sampling, you're likely to have errors in plot. Okay, so how do we decide what will be my sample size? Very simple, two methods I'm going to tell you. Normally in forest department, they use systematic sampling, but I would suggest you to go for the random, and within random, stratified random sampling, because then you can cover all the strata. You don't leave anything actually. That's very important. So what you can decide on that, 0.0001% to 0.005% of the total area you can calculate. Okay, that means you want to do this much because this will depend upon the time and money available to you. You can decrease and increase sampling intensity depending upon your objective. This is another very simple method proposed by Chaco is here. N is the sample size basically. T is the t-test value at 95% confidence level. CV is the covariance square divided by E. E is the error percentage. So with this equation also, for a medium size study area, medium to large size study area, you can find out the sample size. How much area has to be sampled actually. So this has to be done scientifically because you are trying to do some assessment which is based on some. This is how, just an example, how you use this homogeneity map. This is the FCC. This is the vegetation cover type map here, let us say. This is the NDVI map of it. This is the administrative boundary map of it. And this is the density map from Forest Survey of India. So now I know if I zoom in further, let us say I've taken 2 points here. So this point and this point, okay, you can see here. So here it is a brighter color. That means NDVI value is more. This is more NDVI here. This is darker, means less NDVI. So within same forest type, the forest type remains the same, but the density changes. NDVI also has changed. So I will be able to capture all that variability, and then I put this boundary. I can see they are falling in 2 districts. I can put the density. They are falling in 2 density classes. So this is how we prepare the homogeneity map. Based on that, do the stratification. That's what we have done for the entire country in this project, where we had around 22,000 plots identified for this. Okay, there are different, again, approaches. I mean, you can take a big plot like this. Within that, you can have smaller plots like this here, or within this, you can have still smaller plots. It all depends how you want to do. So in forestry, we use multi-phase or multi-stage sampling. Okay, so let us say accordingly we do sub-sampling. First time we will take all, next time only one plot. That's how we do it. This is the concept we implemented here actually. Maybe if you're doing at a state level, you can follow this. This 250 meter is the modest pixel size. Okay, this 250 meter. This is the scene center, pixel center. You can see modest pixel center here. If you go around, that is a 75 to 90 meters, northeast corner, southwest corner, northwest corner, southwest corner, and southeast corner. You can lay 0.1 hectare plots here, here, here. This is called cluster sampling. Okay. Now within this plot, this is 0.1 hectare plot. Within this, I can further divide it. This is 5 meter by 5 meter for shrubs, 2, one in this corner, another is in opposite corner here. And then I have 1 meter by 1 meter, 1, 2, 3, 4, 5, 5 of 1 meter by 1 meter for herbaceous samples. So this is very exhaustive methodology where you can do the sampling of almost all components of vegetation present in one forest type, covering almost all variability, because in one area of 250 meter by 250 meter, you are laying 4 plots. And when you do the mean of it, and you know the pixel value of it, you will get a very good relationship with the satellite data and good for modeling actually. Okay. Let me give you how do we decide on that. Let us say this is an FCC of quickly go through FCC of, let's say, Doon Valley. This is a red color is a sal forest. This is in between the ridge dry deciduous, and this is a mixed white deciduous. All this is valley is sal forest. This is a temperate forest here. Okay, pine and the said other things. And now I have, let us say, vegetation type map. Quickly I will run through here. Let us say this is my vegetation type map. Now I can put the grid here. Okay. I can do the systematic sampling. Every grid I can take 1, 1, 1. I will not suggest you. This is a practice which is followed by the State Forest Department, but I would say we'll go for the. And then you can do the random sampling. You can go wherever you want. Okay, you can do the sampling like this, or you can do the cluster sampling. So wherever you go, take 4 plots. Wherever you go, take 4 plots. Okay, and why you can put a road network here also. Put a road network. So same forest type. If it is closer to road, take there. No need to walk 2 hours, 3 hours. Let's say this is one forest type. Okay, dry deciduous forest. Now this is the Dehradun, Saranpur, Delhi National Highway. So if same forest type, if available near to road, I will take it here. No need to walk down all this. Okay, then I can lay these square plots or point circular plots or transit along a gradient. I can do that. Anything I can do. This all depends upon what kind of a sampling you want to do. Okay, I will not go into more of this. Let's quickly tell you what we do. Please remember, once you have identified the location where you want to go using GPS, take a compass, always look to the north. You can see here this compass, some person is holding it and he is asked to go on another person to exact north. So that's how we direction is fixed. North, from here it will go to east, from here east it will come south, south to again to west. So that's how the whole plot is laid. You can have different approaches, but please be careful. A plot size should be having same bond, equal dimension. I mean, if your plot is square, rectangle, or whatever you are doing, dimensions should be known to me. Okay, now let us say I have now all the measurements done. So at breast height, 1.37 meter. There are many variations in that. We will not go into that. Let us say 1.3 meter in Europe and all that, they use 1.3 meter height for DBS measurement. In India, we use 1.37 meter. Please don't worry about it. This is how we measure. You can see here. So we mark it here, this tree. You can see here, this is a tape, measuring tape. He's measuring the diameter of girth of it. Girth we will convert into diameter. And you can see how it is done. First of all, he marks with a chalk here. You can see he's marking a chalk. This is the height of it. And then some team comes, they take the measurement at the same marked place. And then once this is done like this, you can cross it. You can cross. That means this tree has been already measured actually. This is how we do it in the field actually. These are some of the situations where in normal situation, 1.37 meter is okay. But suppose there are buttresses, there's bifurcations. So if bifurcation is, let us say, less than 1.37 meter, then there will be two trees. If there is a lot of buttresses like here, then please go to the clear bowl. It may be more than 2 meter also, because these buttresses you can't take proper measurement. So these are some variations in the field you will come across. This is how we do the field work actually. Okay, you can see he's taking the branch measurements. So length is known, diameter is known, okay, for each one. And we are fixing these kind of coins below the tree, so that when I go after 5 years or 10 years, I know this tree, number one, this was the diameter and this was the height of it. And this is the species. So I can correlate, let's say 1990 or 2000 or 2015 and 2021 or 2040 or 2050. Same tree can be measured. We know the maintained history of it. So this is just an example of permanent sample plots actually we can do. And this is how we take the measurements in the field of different parts, leaf, branches, debris, humus, all that we do it here. Okay, now let me go to these are some instruments which you will be using. This is called hypsometer to measure the height and slope. Okay, this is called dendrometer. This gives you very accurate centimeter level or millimeter level accuracy you can get with this equipment, but may not be very practical sometimes in dense forest, but open forest this is excellent actually. Yeah, that's how you can see. This is hypsometer. You can use the height of it. This is the dendrometer. We can take the height of using dendrometer as well. Okay, now let us say now we have the measurements. Now I will go for the calculations of it. So as I said earlier, we have now DBH, total height. I have now first choice is species specific volume equation. First choice, calculate the volume or biomass or carbon directly. If not, then use regional specific. That means a volumetric equation of same region, may not be same area, same region. We call them as a phytogeographical region. That's cold desert or hot desert. This is one phytogeographical region. So let us say Sal volume equation is not available. Let's say for Dehradun it is available. For let us say in UP it is available. Let us say for some part in Bihar it is not available. But Bihar is different. Bihar comes from subhumid actually. And this is moist humid. So you can't use it. So region specific equation has to be used. There are state specific general equations are also available in literature. So please use them. Okay. If nothing is available, form specific, form, tree form. Based on that also you can calculate. If that is also not available, genus specific. There's a Shoria robusta. Shoria is genus. Suppose Shoria assamica, you don't have the volume equation. So you can use Shoria robusta equation in case it is not available. If nothing is available, this is called quarter girth formula. If nothing is available, you can calculate the volume of a tree using quarter girth formula. This is the quarter girth formula. You can use that. So let me tell you how these volume equations vary actually. So for Shoria robusta, you can see here, this is for UP. This is the volume equation here. So they take diameter, under root of diameter and volume under root. Odisha it is different. Same species. In UP it is different. For Odisha it is different. For Himachal Pradesh it is different. Teak. In UP it is different. Maharashtra it is different. Rajasthan it is different. Let us say in hills, pinus, cheer pine. UP it is different. Himachal Pradesh too. You can see two equations. So you can understand the site conditions, climatic conditions govern a lot for the growth and density. So please be careful whenever you are trying to assess biomass or carbon, use site specific. Okay, how to generate equations? I will skip here because I told you, you can take a tree borer, put it, pull out, and then you can find out the diameter of it, you can find out the girth of it, try to develop a model out of it. This is a little tricky thing. I'll show you some results which we have done it. This is for the Madhya Pradesh in Madhav National Park. We generated new equations actually. So you can see here, this is for Milusa tomatoes. This is Milusa as a non-commercial species. See, the volume equations which are available in literature, they are basically for commercial species. For non-commercial timber species, they have never bothered actually. So you have to calculate, because now we are talking about science here. We talk about climate change. We talk about carbon assessment. So we generated for Milusa. Gigiphus, this is a bear, Gigiphus xylopyra. Nobody, Lania coromandelica, of course, may be available for south, but may not be for here. Tectona grandis, for that area, it was not available. So we developed our own equation and published it. You can see the publication here also. So what I'm trying to say, you see the R square value is excellent because we have done this sampling based on certain very scientific input and calculated and generated a volume equation for that. Okay, let me just go through in our case study how to use from A to Z now. So this is a case study of Gwalior and Shivpur district in the Madhya Pradesh. So you can see this is MP. In the northern MP, we have taken two districts. So this you can see Gwalior and the Shivpur, and Shivpur is this one is Shivpur, this is Shivpur. We collected data and all that. I have already explained it. So first thing what we did, we tried to correlate basal area versus biomass. So you see the basal area, diameter is mentioned, so I can calculate the basal area. Now diameter is known to me, I can calculate the volume of it. I know the volume, I can calculate the biomass. So you can create this kind of equation. So you can see here, now this is the equation with me. Y is equal to 5.3612 into x. X is my independent variable, let us say diameter. And then I have a beta constant. So this kind of linear equations have to be generated actually. And how to check the error. See, in nature forest, if you go, let me tell you a very basic concept. In plantations, you have uniform crops. The number of plants could be very high, maybe very large number of trees will be growing, but diameter will be very narrow. If you go to natural forest, climax forest, the number of trees will be less, but the diameter will be huge. Are you getting? So that means you can establish a relationship between the number of trees and the biomass, and you can get this kind of curve. And you can look at this is a very good curve here. Look at this curve. I can see this value. That means there is some problem with this. It should have followed this trend actually. It should have followed this trend. See, biomass is basal area is following nice trend. But biomass, when I calculate, see, I see jumps here and there. These jumps, I have to take care of it. I have to check where is the error actually. So using this basal area or any parameter, you can create this kind of error graphs, and then you know, oh, this point is useless actually. I must either discard it or I must find out why it is like this. So error checking is also very important, because then only your confidence level of biomass or carbon assessment will be high. Now we will talk about the spectral modeling. Okay, look at this diagram first. Now this is a FCC of a MODIS data, and you can see 4 dots here. Now these 4 dots are the cluster samples. So within these 4, there are 4 plots. I can find out the volume, biomass, and carbon of each of these 4 plots, and I take a mean of it or average of this thing. Okay, and pixel value is known to me. NDVI value of this pixel I can calculate it. So now that means I know the volume or biomass or carbon in that pixel. First thing, I know the spectral reflectance, let us say in green, red, or infrared, or middle infrared, whatever the band you have in your set from satellite data, I can now calculate. So I have the table of biomass, carbon, then red band, infrared band, middle infrared band, what is the value of it. That's it. Now I can generate a model out of it. So very simple. So we have the pixel level biomass I said earlier. We import those values to the GI in the image processing software. Use any R, NIR, MIR, any or a ratio indices or anything, whatever you have. Develop a model in SPSS or R, Statistica, whatever sort. If nothing is available, you can use MS Excel also. And try to generate a linear function. That's very important. Okay, try. You will have, there are many possibilities of. And then you can use different functions, logarithmic function, exponential function, power function, linear function, anything you can use to develop to get the R square value high actually. If there is a still problem, let me tell you. Suppose this, look at this diagram please here. Suppose one of these plots, no, is at the border. Okay, now suppose it was here in this pixel on the left side. What will happen? Three plots in one pixel and one plot in another pixel. And when you take off mean, neither it belongs to this pixel nor it belongs to this pixel. You get wrong assessments actually. Your correlation becomes very poor. What do we do in such situations? We take the average of 3 by 3, 9 pixels. So I take these three like that. I take 3 by 3 matrix pixel, average of it. So all the mistakes because of the minor variations or GPS location variations are taken care actually. That also is this is last resort actually. I'm telling you the tricks of that to get a good relation. If you get without doing this, excellent. If you don't get it, yes, please use it. Okay, now let us say I have the, I want to extrapolate now this thing. Okay, so this is the modus pixel here. So I have a vegetation type map here. See, I have laid 1 in 1, 3 plots in one type. Okay, and I can have the density map here. Okay, so my density map, type map, and then I get the biomass. So this I can get now a biomass type wise density map also the in that particular area. Now, how do we develop models, let us say? So I have a very simple regression model. So we used linear, logarithmic, power, and exponential functions. So we have taken 4 months or 4 season data, February, May, October, and December. And we use, let us say, red band. So first of all, we are trying to relate with the red. Red is a very strong absorption and related to the amount of biomass. Okay, so I have, let us say, 78 are my observation points. So my R square value is coming, let us say, best in this is around 5.2. I think this is 5.35 is the best R square value with the linear function in the May month. No, this October month is coming better. Okay, so this. Now I do with the infrared band, let us say. Okay, I do with the same thing, linear, logarithmic, power, and for all these 4 months. That's why I was telling you, multi-season data is very important. So I take and then I get some, let us say, constant here. Now, if I use NDVI, let us say, simple NDVI, okay, not advanced or ABI or EVI and all that. Just you can see here, with the logarithmic function, with the October month, I get 7.57. Sorry, 0.757 R square value, which is highest among all these things. That means out of how many combinations, you can see here we had 416 and we are around 48 combinations here in the 3 and red, infrared, and I found with NDVI, it gives me the best, that to logarithmic function, not with the linear function. If I want, I can now find out, I can run the model with linear as well as with the logarithmic function to find out the predicted biomass value of that pixel for which you already have the field observed value. So you know the errors actually. Is it coming very close to it, exactly same or different? And then you can decide which model is going to be good for me for my study. So you can see here we have taken this 0.755 NDVI. So plot like this. So October NDVI with the biomass turns per hectare or turns per pixel it should be actually. Okay, and then we can lay like this. You can see here, and that's how I now get an equation. Now this equation I can use and put on that. So my x is what? My x is NDVI. So I just take NDVI log of this and run it. I'll get a very good biomass bar. So you can see here now this is the photo biomass map of the entire area here. This is, let me tell you, this is the Gwalior district here. This is Shivpur district. This is Shivpuri district. This is covering a huge area of Madhya Pradesh. Okay, we actually have extrapolated to larger area. It may be around 500 or maybe 200 kilometer or maybe more than 500 kilometer by 500 kilometer region actually. So you can see here now this is the Madhav National Park here, this range, and you can see how the vegetation is varying and how the biomass is varying and how the carbon is varying. So you can see here the protected areas. I mean, here you can see the protected areas wherever ridges, because the plains have been already cultivated or they are very highly degraded. Only hills where steep slopes are there, where boulders are there, stones are there, people are not able to grow crops there. So there you find the good biomass, good trees covering. So you can see here now this is geospatial map of the phytomass of that particular region. So you can similarly convert this into carbon also, and then you can try to convert this. This is predicted value from earlier observations. We got the field value. So observed value versus predicted value. So this is predicted value on this side. This is the observed value, and you can say here R square is around 0.84. It's excellent, I would say, in such situations. Okay, so that means the model which you are going to produce out of this equation is going to give you reasonably good result out of it. So this is for MODIS data, and MODIS data 250 meter by 250 meter. In 250 by 250 meter in field, you may have several things actually. You may have good forest cover, you may have blank areas, you may have rock crop out, you may have grasslands, you may have some part of river in that one, some part of barren in that one. So you can imagine, in spite of all that variability within 250 by 250 meter of the pixel in MODIS, we are still able to obtain 0.84 R square value. That means the model is very good. Suppose I take Landsat TM data 30 meter or IIRS list 23.5 meter, or I go for high resolution data, let us say 6 meter or 5 meter. I will get better results provided I am able to find out where my plot has been located. Locating your GPS error should be minimum actually. See, if your error is even 1 meter, your biomass is for 1 pixel and your NDVI is for neighboring pixel, you will never get a good result. So try to be as precise as possible in taking the field measurement so that error is minimum in that. So that's how we have now the carbon map, and this is nothing. If you can see, this map is almost same map. What we have done is here, because I know that the carbon content in a phytomass is let's say 0.475. So just multiply in GIS. So you take this map, and into 0.475. That's all. You get a carbon map. So you are now able to get the carbon map of the entire country. You can now quantify. So this is telling the range here. This is telling the range, but you can find out the quantified total pixels you can count out in either you use the image processing software or GIS software, and you can find out the total carbon present in the area. That's how this is for time one. Let us this study. I think we carried out in 2008 or 6. Now, if you go, situation might have changed because either vegetation might have improved or degraded. So in this equation, you can use it, but results will be different depending upon the condition of the vegetation found in that particular area. And then upscaling is also required. See, now this is pixel level. When you are working for a small area, pixel level biomass is a small file which you can share, put on net and all that. But suppose you are doing a country level carbon assessment or biomass assessment, then if you put a huge, which is in terabyte, let us say, data, you will not be able to file actually. So we can generalize it. So this is a method. What we can do is, suppose this is another study which we did in the Surat district. So we can lay 5 kilometer by 5 kilometer grid on it, and then can find out the area in each grid under each class, how much of area is and how much of biomass is there in each of these, and put that value in that. So instead of having the value which was there, now you can total it up within 5 by 5 kilometer. How much of biomass is there? Let's say 500 tons. Okay, you put the 500 tons in that value. Then you get a generalized map. It is quite similar to one, but it is quite generalized. The file size decreases tremendously. You can put on net, people can visualize it. This is good enough for visualization, for planning and management. Ultimately, when you do the implementation, you have to go to the ground. That's the reality. It's your one-to-one. So this gives you the idea, okay, what kind of activity has to be done, afforestation, reforestation, whatever you want to do based on this poorly stocked or good stocked. If it is good stocked, maintain it. If it's poorly stocked, improve it. All that kind of management decisions can be taken. So you get this kind of map. For this map, we can get this kind of. So this looks very generalized map. But this is good enough that it tells me, okay, this area is high carbon map, this is a low carbon map. This is basically agriculture, all this low is agriculture area. So biomass and carbon map. Only legend will change, legend will change, that's all. Rest is same. Okay, friends, so this is all from my side. I think it has been a little lengthy, but I thought let me expose you to the whole game of the biomass and carbon assessment in the forest ecosystems. So thank you very much.